Okay, so hopefully we're back in. I'm not sure what happened there. Zoom kind of did a weird blip on me and it looks like it basically restarted. So hopefully we're in good shape now to get back into the class. So yeah, we were talking about microbial culturing last day, and we were talking about McConkey agar and blood agar, and I had a few other things to share with you regarding topic 7. So one of the things I want to show you is this here. There are many types of these agars out there that are differential or selective. This company here, Hardy Diagnostics, I think they're called, specializes in these kind of things. And I saw one of these products here. This is really quite incredible. These are these agar plates where you put your urine sample on it, and 1/2 the plate is selective for gram negatives, 1/2 is selective for gram positives. And then there's some sort of colored indicators in there so that the clinician can take a look and see all sorts of different organisms. And so you can see there's the scheme, right? So Enterococcus is teal, Staphylococcus epidermidis is white, and so on. So I thought it was kind of an interesting thing to show you. Can we buy these and do this at home? Haha, nice question. I'm sure Hardy Diagnostics will sell you. They're a company interested in making money. If you want to look them up and buy things from them, go for it. Check on Amazon too, you never know. So I wanted to mention a few other tests that, there are many tests, by the way. There's hundreds of tests, but I wanted to show you some what we call, I guess, biochemical tests, where what you're doing is you're taking the organism and you're looking to see if it has a certain biochemical property. So here's one, for example. This is called the catalase test. So if you take a look, catalase is something that uses this compound here. So this is hydrogen peroxide, and hydrogen peroxide is a relatively reactive compound, and it can get broken down into water and oxygen. So a number of organisms do have the enzyme catalase, and the test is super easy. Basically, you take a little bit of culture, you put it on a glass slide, and you add hydrogen peroxide, and you look for bubbles. I actually did this 2 weeks ago. I could not get nice pictures. It happened so fast. By the time I got my camera out, it had stopped bubbling. Somebody here on the internet found a nice one. You can see we have the difference here between Streptococcus, which is catalase negative, means it does not produce bubbles, and positive, which means it does produce bubbles. So this is a good test for Staphylococcus, right? Do we have Streptococcus? Do we have Staphylococcus? Do the catalase test. Super easy. I'm not sure if there's somebody who's trying to ask something or whether they just have their microphone unmuted, but either way, either muted or ask a question, and that would be great. So there's other tests. I want to show you the coagulase test. So the coagulase test is where the organism actually binds something that's found in our connective tissues called fibrinogen. And so what organisms do that? This is specifically a test for Staphylococcus. So if you take a look here on the left, so this one here is negative, and this is for Staph epidermidis. And if you don't know which staph you have, you throw in this fibrinogen, and it will actually break this down and make these clumps. And so this is for Staph aureus. So these are like quick and dirty tests. And, you know, like I said, you can kind of think you've got an organism. You start off with your gram stain, and then maybe you start going through other tests to try to sift out what exactly it is that you have. These are very fast acting tests, very easy to do. And when I teach a microbiology course with labs, we do some of these tests. And like I said, they're very, very easy. So I'll just show you one other test here, urease test. There's a whole bunch that are like this. I'm showing you this test. I won't ask you about the urease test, by the way. But I'm showing this one to you because there are a lot of tests like this where people have found a way using some chemistry to give some colors. And colors are great. You add your bacterial culture to a tube or tray or whatever it is, and you know, you have a scheme, and the scheme might say this is going to be purple if it's positive and pink if it's not. So assuming that you you know, you're not colorblind or anything like that. Some people are asking about notes for topic 7 and 8. The notes are there, the PowerPoints are not there for topic 8 and later, as far as I know. So please take another look there. I'm just finished this PowerPoint this morning and haven't had time to actually post it for you. I'll be able to do that after class. So there's companies that have capitalized on this, and they've made these, it's kind of like a big sheet, and it has these tiny little chambers where you can add the culture and then look at a scheme and see exactly what organism you have. These are really interesting tests. Again, you know, these are for clinical diagnosis. You can see this is testing a whole bunch of things. I don't recognize all of these, but we have alcohol dehydrogenase. Oops, my pen loves to do that. Lactose dehydrogenase. So do they digest lactose? That's an indole test looking for tryptophanase activity, etc. And so, like I said, you look through a scheme and it would match certain organisms. So somebody's asking, I think there's a question about what do you need to know for the midterm 2? Well, we'll get there. Definitely the catalase and the coagulase test. And I actually have a case study question here for you guys that we'll look at right now. So you can see here's a case study. You've got some patients in the hospital and these skin infections are going around, and this is for something called MRSA. So we're going to start talking about MRSA a lot. This is a super important organism, and it's kind of like the #1 superbug. So you can see MRSA, it stands for multi-drug resistant Staphylococcus aureus, or sometimes we say methicillin resistant Staphylococcus aureus. So we'll talk about that in a couple of topics from now in a lot more detail. But this is a strain of staph that is, like I said, it's the #1 superbug, can cause skin infections, and it can pretty much infect any part of your body. So if you're in a hospital and you're getting invasive procedures such as surgery or catheters, it can infect your blood, it can give you pneumonia. So this is a very dangerous organism to have because it's also hard to treat. Now we can't go along and just, get everyone in the hospital. So quick and dirty thing to do is to test everybody for MRSA, right? And so, you go around and swab and test, and these things can be just put on some Petri dishes. So 1 Petri dish per patient, that's not too bad. And maybe do a few other tests to kind of, see what it is that we have. So here's some of these tests that we talked about, and we can go through this and kind of figure out what this should look like in terms of these tests. So growth on MacConkey agar. So none, right? So remember that only gram-negatives grow on MacConkey agar. It's a selective media. So growth on blood agar. Probably if you're looking for MRSA, you probably wouldn't waste the MacConkey agar. You'd probably go right to blood agar and streak it out on the blood agar to see what it is that you have. So someone's saying that their Moodle ends at topic 6. So I will take a look at that after class. So sorry about that. Growth on blood agar. So yes, Staphylococcus grows on blood agar, and it is beta hemolytic. So that means we're going to get the halos around the colonies. It's a little more weakly beta hemolytic than some other species, but usually it ends up being kind of yellow as opposed to greenish, and those halos around the colonies. So gram stain, gram-positive staphylococci. So I think I had a similar question like this last year on the midterm, and give me details, right? So when I ask you what it looks like under a gram stain, purple, that's kind of half of the answer. Gram-positive, I understand that gram-positive means purple, that's fine as well. But what are you seeing? Are you seeing purple rods? Are you seeing gram-positive rods? Are you seeing gram-positive spheres? So this is the full answer, right? Gram-positive staphylococci. You can use different words to describe that. You can say purple clusters of spheres. I'm okay with that either, but do make sure that you are complete. Catalase test, we just talked about that. Staphylococcus, both Staphylococcus that we're been talking about are positive for the catalase test, which means you're going to see bubbles. In the coagulase test, you're going to see the clumping, and that's going to be for Staphylococcus aureus. So this may not be necessarily the end of the story. Sometimes you might also do antibiotic tests. And why would you do that? You just want to see what strain of Staphylococcus you have, particularly if you're looking for something that's drug resistant. You're testing to see which drugs are actually going to work. So hopefully that makes sense, right? You can't just start giving people antibiotics and then they're not going to work and you're going to prolong their stay and all that. You want to do that that test for MRSA to see what they can actually be prescribed. Okay. So that is it for topic 7, which, like I said, it kind of blends into topic 8. Topic 7 was about culturing and growing these things. Topic 8 is what you're going to do with those cultures and how you're going to identify them so that in theory we can treat people. I see there's another question. Somebody's asking about hydrogen peroxide on a cut and it bubbles. There are probably bacteria present, yes, but you also have catalase. You also produce that enzyme. I'm not sure if it's in your skin or if it's in your blood, but that's your enzyme that's breaking it down and making the bubbles. Okay, so I actually showed you these slides before and said that we can collect our sample. Some diagnostic methods are going to depend on culturing. So we're looking at growth characteristics. Does it grow on blood agar? Does it grow on McConkey agar? What color is it, etc. Does it have any metabolic activities? Is it catalase positive, coagulase positive, and so on? And we're going to talk about drug sensitivities and things like that in a couple of topics from now, so we'll get there. And I showed you, I did some swabbing, and those were our results. We have some colonies, different shapes and sizes. And if you've done enough of these things, I can usually look at these plates and find you Staphylococcus pretty easily. Staphylococcus is, I'm guessing this here's Staphylococcus, probably this one here. This one here is a little harder to tell from the image, slightly different browner color. It could be E. coli. I wouldn't know until I checked it. If I had the plate in front of me, I might be able to tell you. E. coli tends to be a little shinier, a little bit browner, and so on. The sponge, I have no idea. There could be anything on that. Didn't want to touch it. The toothbrush, again, I'm not sure what those yellow things are. I'm not as familiar with oral bacteria, but you can see there's some yellow colonies on there. A lot of that is probably Staphylococcus. It grows really, really well in Petri dishes. Same thing with this one here. You can see I'm seeing a lot of Staphylococcus all over these. It's not quite white, but it's close to white. This one here has some kind of, those blurrier colonies, and that's probably some sort of bacillus. So we don't necessarily know until we do the gram stains and things like that. So that's what we're going to talk about today. In fact, we're going to talk about a whole bunch of things. So we're going to talk about microscopy. So I have a lot to say about microscopy, and not sure if we're going to get to those other parts, but we want to talk about serological, immunological characterization, and also how genetic tests work as well. So that was topic 7. Now we're going to look at topic 8. Okay, so I think I showed you this exact slide before when we were talking about how big things are, right? So this is just kind of a reminder. The human hair is a good place to start because that's about the smallest object we can see, around 100 micrometers. Lower than that, most people, their resolution of your eye is not any better than 100 micrometers, so anything skinnier. So that's going to include all of these microbes. So most protozoa and fungi are smaller and skinnier than human hair. Notice I said most. There are a few that are larger. You can see right here there's paramecium. So paramecium are large enough to see with the eye, but they look like the smallest speck you can imagine. So not much useful. We're not seeing a lot of detail there. Bacteria are smaller than that, and viruses are smaller than that. And if you look at the bottom of this slide here, we have, you can see the different types of microscopy. So this one here is a compound light microscope. So you can see for the most part, you're not even seeing viruses at all. You need an electron microscope to see those things. And sometimes in no detail. So we're going to talk about both light microscopy and electron microscopy and a few other things in between and a few tricks we can do to improve our images. So if you think about microscopy, there's kind of three things that we need. One, a specimen. Two, some sort of illumination source. That's an eye. And three, some sort of lens, and that's a basic microscope. So you can see there's my object there. I've got my lens. I'm going to illuminate it with probably some light, and then you get a large image. So a magnifying glass is a very, very basic microscope. And in fact, we've seen something that's like a magnifying glass when we were looking at Van Leeuwenhoek's fancy microscopes. So that's what you can see on the left there. There's Van Leeuwenhoek's microscope. Basically, like I said before, it's just a lens there, and then he has an apparatus to hold it and adjust the specimen so it's a little closer or a little further to the lens so you can focus it, and then he's just using light. Hook's microscope there is in the middle. It's a little fancier. It's a compound microscope. Compound microscope means it has more than one lens. You can see he's using an oil lamp to try to look at his specimen. And most modern microscopes are a little bit like that one on the right. And what you're seeing here is we have a compound microscope, but it has different objective lenses. So I can zoom in and I can zoom out and see the image a lot clearer. So there's Van Leeuwenhoek with his microscope and looking at some specimens there. And like I said, it's not much more complex than a magnifying glass. So most people nowadays are using some sort of compound microscope. You can even buy cheap ones at toy stores and Walmart. And we'll talk about what the difference might be between the cheap ones and the more expensive ones. You can see this one has a few more bells and whistles than just lenses. There's a prism there that kind of bends the light and makes it more ergonomic. Some of them have two eyepieces, some of them have one. This one has this condenser lens. So the condenser lens focuses the light on the slide. So, you know, there's a lot more things that you can do with it in terms of, you know, giving better fine control and those kind of things. So I want to talk about what we can do to get good images, right? And there's some different variables around that. So I think I already mentioned magnification, right? So there's different lenses. And I had at some point, I did have a plastic microscope at one point. I wonder what happened to it. I guess I probably got rid of it because I have better microscopes now. But it did have different objective lenses, and you could zoom in on things. Okay, but think about zooming in, and I'm sure some of you have done this with your cell phone, right? You can zoom in on an image, but there's just a point where the image, yeah, it's bigger, but it's not clear, right? So that's what resolution really is. Resolution is the clarity of an image, or if you want to be more precise, it's the minimum distance of two indistinguishable points. So I'll show you an example here. And some pictures. So there's some photographs of some cells or some object anyway, and you can see that the magnification is the same, right? So, but the resolution is very different. So the one on the left, you can see the resolution is about 2 micrometers, and I can't necessarily make out the individual cells. I can sort of, you know, maybe use my imagination and picture where they might be. But the one on the right, look at that, much, much better. So when, you know, with resolution, often we'll give a value. We'll say the resolution is, let's say, you know, 10 nanometers or 5 millimeters, or in the case of microscopy, usually we're using micrometers and nanometers. You know, something that's useful. Bacterial cell is about 1 micrometer. So if you want to see E. coli, we need to have a resolution of about a micrometer or a little bit better. So hopefully this is obvious that microscope B is the one with the better resolution. There's a cartoon for you. Resolution is clarity. So there's the far side, and you can see there's some amoebas or some sort of unicellular organisms, and they're looking at family photos on some sort of slideshow, and they say, hey, wait, that's not Uncle Floyd. Who is that? Oh, maybe it's just an air bubble. So Thought that one was kind of cute. Okay, so sometimes we can get good sharpness, good resolution. Sometimes we can get good magnification. Then we can't see our things very well. So kind of the last aspect of something that helps us to improve our image is something called contrast. So if you take a look, this slide here demonstrates contrast very nicely. On the left picture, the cells are very similar color to their background. So contrast is kind of the sharpness between an image and its background, right? And so you can see here that we can see these images very well with this gram stain. So staining is what we're doing to improve contrast. So staining at least is nice and cheap. If you want to improve these other variables, resolution in particular is something that's going to cost you a lot. So let's go back to that Walmart microscope or older cell phones, and they talk about, you can get a certain amount of magnification or a certain amount of zoom or something like that, but the images, they're just terrible, right? So what is the difference between that Walmart microscope and the the $7,000 one I have in the biology lab? So obviously we're looking at much of it made of better materials. You've probably got better focus knobs, et cetera, et cetera. But the big part of it is actually the lenses. And how do we improve resolution with lenses? You get better materials. So you probably know that the Walmart microscope is probably going to be made out of plastic. We can make plastic lenses. Some of you may even have eyeglasses with plastic lenses. There are good plastics, there are cheaper plastics. So the more expensive plastics make better lenses. And if you want better than plastic, you're going to use things like glass is better than plastic. So many eyeglasses are made of glass. And if you want even better than that, you're going to use quartz. And that will cost you big time, right? Like to replace one of my objective lenses, I think that's $1,200 for one lens, right? It's made out of quartz, but you're getting much, much better images. There's other ways to improve resolution as well, but that's kind of the big one is, you get yourself a good quality microscope in terms of the lens material. Here's some more pictures for contrast. Like I said, contrast is easy to improve. You just use stains. Stains are cheap. Well, most stains are cheap anyway. You can see there's a nice picture there where we can see the cell in two different pictures, and the stained one is it's just so much. It pops out better. My eyes can find different objects better. The nucleus is so much more obvious, etc. I know I have some other pictures here. Lots of nice pictures to show you today. There is a stain of Giardia. And, Giardia is such a cool organism to look at with those two nuclei and those flagella. They look like little aliens. Somebody's got to make a good science fiction movie where we're getting invaded by giant Giardia, and I think I would go see that one. So let's talk about that gram stain, okay? And I have quite a bit to show you about the gram stain. Very important stain. It was named after a guy. His name was Graham, Hans Christian Graham. And this is 1800s, right? So people have discovered bacteria, they're looking at them, and he discovered a way to stain them and found out there seemed to be two different types of bacteria, the gram positive, which stain purple, and the gram negative, which stain a pinkish reddish color. So I'll show you some gram stains here in a moment. I think I have a reminder here of what is different between gram positives and gram negatives. So you can see I have the gram positives with the purple font, remind you that they stain purple, and they have the big, thick peptidoglycan layer. And the gram negatives have a thin peptidoglycan layer and an extra membrane, an outer membrane on them. So I want to talk a little bit about the procedure, and then I have a little demo video I'll show you of me doing a gram stain. So let's talk about what's going on in the gram stain in a bit more detail. So the first thing you do is you add this crystal violet. And so crystal violet, maybe I'll represent it as a V. So it's a positively charged molecule. And at this stage, it kind of sticks to everything. So if I were to look at my slide here, I would see all my cells would be purple. Some of the purple would probably be on the glass as well in the background. But sometimes people will do this. They do a very quick and dirty step because they just want to see their sample really quickly and they just add the crystal violet. But a full gram stain isn't a big deal. It's not a very long procedure. So the next thing is you add the iodine. So iodine is a negative charge. So here's my iodine. And so you can imagine the iodine and the crystal violet is now, it's starting to circle my charges so you can see them. I'm also drawing lines. They kind of, get together and they form these little complexes. Or actually not little, see my complex is getting quite large. So now what we have are these big complexes. And remember, we have that peptidoglycan layer in the gram-positives, and these complexes will actually get stuck in there. The gram-negatives, the crystal violet is kind of still loosely associated with the outside of the cells, and that's about it. It's not getting stuck there permanently. You can wash it off with enough washing relatively easily, and that's actually what the next step is. It's a washing step. So there's different recipes for the washing step. People use ethanol or acetone. It doesn't really matter. You can wash the cells, and the alcohol will dissolve these complexes very easily, or at least it will do so in the gram-negatives. In the gram-positives, remember the iodine has gone in there, and these things have turned into these large complexes, and they get stuck in the mesh. In the gram-negatives, this will also kind of wash away a little bit of the outer membrane, and all that gram-negative, or all that crystal violet stain will go away. So now you have purple cells and kind of transparent cells. So we want to see the gram-negative cells. We add the safranin. That's a reddish pinkish color. And so this sticks to everything. This sticks to everything at all. In fact, you'll see some of my gram stains. You see a pinkish glow to things because it just sticks to everything. So why are the gram-positives not pink? Well, because think about it. If you had a dark purple paint and you had a pink paint and you mix them together, you still get purple. You just can't see it so well. Sometimes they look a little pinkish. Whereas the gram-negative cells, they look nice pinkish reddish color. So I'll show you a demo here, and then I'll show you some pictures here in a moment. Somebody's asking about the word mordant. Mordant means fixative, right? So it's fixing the crystal violet into place, right? So if you ever do any fabric staining or dyeing or anything like that, you might be familiar with that word. All right, so here's my video. I tried to trim it down to just 2 minutes because the whole procedure took me a lot longer, but I wanted to give you a demo and show you how this works. So I'll play this for you here. I just want to make sure that the volume is going to go through. I think I have to click something special to make sure the volume works. So here goes. And the first step, I'm going to add the crystal violet. So what I do is I add the crystal violet to the slide, put it on top of the bacteria, and let it sit there for 1 minute. At the end of 1 minute, I'm going to wash off the excess stain. And what I'm doing is I notice I'm flipping the slide upside down. I don't want to have the water hit the bacteria directly because that will blast the bacteria off the surface of the microscope slide. So what I'm doing is I'm putting the water on the other side, and it's going to gently curl around the glass and wash away the excess stain very gently. So once I'm done that, I can dry it off on a piece of paper towel by just tapping it, and I'm ready for the next step. I'm going to add the iodine onto the bacteria and let it sit for 1 minute. So I'm going to add the ethanol on there. I don't want to leave it much longer than about maybe 20 seconds. At the end of 20 seconds, I'm going to let the ethanol come off, give it a quick rinse. Take a look at the slide. If there's too much purple on there, and I think our slide is actually looking pretty good, if there were too much purple on there, I would do another ethanol wash step. So very similar like the other steps, you put the stain onto the bacteria. In this case, I'm not going to leave it quite as long as 1 minute, maybe 30 seconds to 45 seconds. I'm going to wash it one more time. So now I'm going to let this microscope slide dry, and then we'll look at it under the microscope. Okay, I hope you enjoyed the little demo there. I actually trimmed that down from a much longer one that I had made for another class. I was just trying to keep it kind of 2 minutes in duration so you can see a lot of cuts in there. But hopefully you get an idea how that works. It's a pretty simple, procedure, and it's pretty easy to do in undergraduate classes. Someone's asking again about the mordant. So mordant step, yeah, it makes the crystal violet stick to the gram-positives in particular. Can iodine kill bacteria? Yes, it can. Yes, it can. So these bacteria on this slide are dead, by the way. They're dead at the end of this procedure. They're dead before the procedure. Before that step, you heat treat them, which probably kills them anyway. And just, there we go. There's some gram-positive and gram-negative cells. And so the gram-positives come out purple. We've already seen anthrax. We've already talked about staphylococcus and E. coli and Neisseria gonorrhea. So, you know, you can see I'm showing you these organisms in particular because they're all very different. So sometimes with the gram stain, it's been around since the 1800s, and it's still one of the first tests done because often, in many cases, you can identify the organism very, very quickly. So anthrax, for example, if you've seen that on your slide, it's pretty obvious what it is. And then you want to figure out where you got it because you don't want it. So shapes and arrangements, we can look at these things too, which are going to be helpful. Do you have a coccus? Do you have a bacillus? Do you have a diplococcus? If you have a diplococcus gram negative, you've got some sort of Neisseria, either gonorrhea or meningitis. So, you know, that's an important one to know, right? Do we have Staphylo? Do we have Strepto? And there are different organisms that fit in those categories. You see, you have a couple other up there. We have Vibrio. Vibrio means a bent rod. And Sporillum, or sometimes spirochete, is used. I'll spell that out for you. Spirochete is sometimes used to describe ones that are really swirly. You know, that includes organisms like the one that causes Lyme disease or also syphilis. So those ones are good to know. And I'm going to make a flow chart for you in a moment, kind of looking at all these things we've been talking about, all these tests, and trying to put them all into one flow chart. So make sure you have a piece of paper ready to go and a full sheet, and if you have a big one, even better. So some of the organisms, like I said, you can add those shapes and arrangements together. So we can have diplococci. Diplococci include, as I mentioned, Neisseria gonorrhea, and sometimes Streptococcus comes out between those. So someone's saying, is it hard to distinguish Bacillus and Vibrio under a microscope? Sometimes. It depends on the organism and the growth phase. There's one organism that I have in the lab. What's it called? Ribosporillum. That one is actually a spiral-shaped organism. But at different times in its life cycle, sometimes it's really swirly, and then other times it looks like a barely bent rod, almost looks like a Vibrio, and other times it actually looks like a rod. So sometimes it kind of depends on the age of the culture too. These things get a lot easier with experience. If you looked at as many gram stains as I have, you know, it's pretty obvious what you're looking at after a while. Streptococci, so that includes the Streptococcus organisms. There's a whole bunch of them. The two that we need to know in this class are Streptococcus pyogenes and Streptococcus pneumonia, although there are many other Streptococcus organisms out there. There's also Staphylococcus, and this is actually my photograph. You know, this one was really disappointing, the photograph they had in the textbook. I don't know why. It's very easy to get a good Staphylococcus photograph, but you can see purple there and the clustered spheres very easily. And then Bacillus, for some reason they didn't have Bacillus, so a nice little display in the textbook. So I made this one here for you. And so some organisms have the name Bacillus as their genus. Unfortunately, these are very, very common, and there's many organisms, particularly in the gram-negative range, that are Bacillus, and so it's a little harder to tell what you have. And so we'll take a look at this flow chart here in a minute. So here are some of the organisms we talked about. There are a few others. I'll probably put a few others in the flow chart, but this is a good place to start for which organisms to know. I will have on the midterm, so here's another midterm tip for you. I will have on the midterm at least one multiple choice question that is going to show you some gram stains. And it'll say, you know, which one is wrong or something like that, right? You know, so you look at that and you see one that's you know, maybe you see some gram-negative bacilli and it says streptococcus, and you're like, wait a second, streptococcus is gram-positive. That should be purple. Although what I am going to do on the midterm, I know some people have color deficiencies in terms of they can't necessarily see color, so I won't be looking that you can distinguish between purple and pink just in case, but you do need to know the shapes. Someone says, can the gram stain be compromised if the fixation agent interferes with the peptidoglycan cell wall? It's pretty easy to compromise gram stains. There's a lot of ways they can go wrong. You can wash too long with the ethanol. You can leave the saffron and red on too long. As I mentioned before, sometimes the age of the culture will affect things, and then it's a little hard to tell, is it a purple or a pink? You know, the most commonly stained organisms, usually, you know, if you've done enough gram stains, you have an idea of what you're doing. But like I said, if you kind of follow that recipe, some people think you should leave the saffron on no longer than 20 seconds. Some people say 45 seconds. So there's a little bit of an art to the whole thing, a little bit of art to the interpretation. And like I said, certain types of cultures, depending on the age of them, may not stain properly. But for the most part, you know, I do this with a lab for my biology 107 students, and I would say a good 85% of the gram stains turn out nice. The biggest errors are people you know, sometimes people have way too many cells on there, so it's hard to see anything. And I would say the decolorization step is the other issue that people have. Sometimes they use way too much ethanol and they let it sit there for minutes, and by then all your gram negatives or your gram positives are starting to lose the purple color. Okay, so I think I have my flow chart is on the next slide here. So what I'm going to do is just switch to the whiteboard, and I'm going to draw a massive flow chart for you. Well, you can probably fit it on one sheet of paper. You may want to have two sheets of paper, one for the gram negative and one for the gram positive. Somebody's asking, are there no gram positive Vibrio? I'm not going to say no, but I can't think of any. So that's a good question. At least something that's medically relevant, I can't think of anything. So here's my flow chart. And like I said, you may want to have one page for gram positives and one page for gram negatives, just depending how big your writing is and all that. So this is where you're going to start. You're collecting your sample. Let me see here. I'm going to get a purple pen for the gram positives. There we go. So remember that gram positives, they stain purple. That barely looks purple. But maybe it's purple enough. And maybe I'll also put no growth on McConkey agar. Okay, so the gram negatives, I'm just going to put the note of what color they stain. So they stain pink, red, kind of just depending on how much stain you have, whether you get pink or red. Okay, so we'll come back to the gram negatives in a minute. Okay, now, like I said, I'm going to try to fit everything on here, and hopefully I can write neatly today. So you do your gram stain, and then you have all sorts of possibilities. And I'm going to break this out into 3 possibilities. So let me just put that over here. So 3 possibilities. Well, there are more, but the ones that we care about in this class is we have the streptococci. There we go. And I'll draw you a little picture so you can remember what they look like. So remember, strep means there's a chain, and cocci means they're little spheres. So there's my streptococci. Option 2, we might get some staphylococci. Okay, so staphylococci are, of course, also spheres, but this time they're bunched up like a bunch of little grapes. And then the third one, we have bacillus. And hopefully when we have bacillus, we don't have anthrax. Okay, so bacillus, oh boy, this is a little harder to draw. I'm trying to draw a rod and it's just kind of looking like a funny oval. Okay, there are my rods. Love for it to be a little neater, but it's really hard with this pen. Okay, so I guess I'll just keep the purple ink since we're gram-positive, so why not? So let's talk about those streptococci, okay? Just underline those because we have 3 categories. So remember the difference between the streptococci and the staphylococci was the catalase test. So the streptococci, remember, were catalase negative. So that meant there were no bubbles with the catalase test. The staphylococci are catalase positive. I have nothing to say about the bacillus for that because some of them are catalase positive and some of them are catalase negative. So no note for that. And it doesn't matter, they're different shape, so pretty easy to tell right from the gram stain. Okay, so remember the streptococci, so I'm just going to move this over a little bit. Just makes it easier for me to write. You can put these things onto blood agar. And we can look for hemolysis, right? So remember we have 3 types of hemolysis. We have the alpha hemolysis. And alpha hemolysis, remember, was partial. And the organism that we're interested in here is Streptococcus. So I'm not going to write out the full Streptococcus, just S. Pneumonia. Okay, beta hemolysis. Beta, that's my beta. And so that's full hemolysis. And in this case here, we're looking at Streptococcus pyogenes. I think somebody has their microphone on. Here's some ruffling. Okay, and one more is the gamma hemolysis. And the gamma hemolysis, there's none, so no hemolysis going on, no halos around the cells. And here we're talking about various Enterococcus organisms. So by the way, I'm not going to ask you about Enterococcus on the tests. You know what? I can't cover every organism. I'd love to talk about Enterococcus, but it's just not important to this class because in terms of the examples I have, it sort of covers some of the same examples. But I'm putting here for completion, so we have an example for each alpha, beta, and gamma hemolysis. I know I'm running out of space, but I'm going to put one other little note here about Streptococcus pneumonia. The Streptococcus pneumonia has capsules, and I'm going to show you what a capsule test looks like after we get through this. So there's the Streptococci. Okay, so what are we doing now with the staphylococci? So as you remember, there's 2 staphylococci that we care about the most. That's Staphylococcus epidermidis and Staphylococcus aureus. And in that case, we want to do the coagulase test. So the coagulase test, remember, that's where we're looking for the clumping with the fibrinogen or not. And if the coagulase test is negative, so no clumping, then probably what you have is Staphylococcus epidermidis. If it's positive, so you're going to see clumps, then you probably have Staphylococcus aureus. Okay, I got to slide this over a little bit, so I apologize if you didn't get all that with the streptococci. I just need a little bit more space. Maybe I can zoom out a little bit. There we go, that's not too bad. So one other thing, and we mentioned this already, with Staphylococcus aureus, often the next step is you're going to do some antibiotic susceptibility tests. Because you want to know, what can we treat this thing with? Antibiotic. Oops. I have an eraser here somewhere. Where is the eraser? I gotta figure out the eraser function. There it is. Antibiotic susceptibility.
I have a question.
Sure.
So is this antibiotic testing just for the aureus one or for all of them?
It's most commonly done with Staphylococcus aureus because there are issues with it, right? A lot of the other organisms, you know, if you have pneumonia, you know, they just go right to the prescription in most cases, right? Unless you're an older person and maybe you've had pneumonia a lot, then they might suspect some sort of resistance. But, you know, that's often the case. Same thing with strep throat. Often they go right to penicillin unless you have issues and they suspect resistance. There might not do the testing. Staphylococcus aureus, very common for all sorts of drug-resistant versions out there. And the biggest concern is MRSA. So they're going to often do the testing. Not always. Sometimes they just throw a really strong antibiotic in there and hope for the best. Kind of depends on the situation, really. Okay, so we have the last gram-positive group are these bacillus. So I'll see if I can write that small. If not, I'll just squeeze it over and maybe the gram negatives will have to be really far to the right. Okay, so bacillus, there's unfortunately there's a lot of different bacillus organisms out there. And this is where it gets complicated. So we have the genre of bacillus. Right, so that includes anthrax. That includes a few other organisms, many soil organisms like Bacillus subtilis. I think what I'm going to do is just move this over a little bit so that I can fit everything in there. So what can we say about bacillus is that they are aerobic. So that's a big one. And they're spore formers. So those are two things that are really easy to test for. If it's aerobic, does it grow with oxygen? Yes or no? If it's spore formers, it's very easy to test for spores. You can take a sample and you can heat it up for a bit and then put it on a plate. And if you know, things that aren't spore formers aren't usually going to kill them. I see there's a question someone's asking about, do broad spectrum antibiotics work to eliminate Staphylococcus aureus? Yes, they can. Again, it depends on the strain. And I think that's often the strategy is somebody will prescribe a broad spectrum antibiotic and hope for the best. Now, there are many cases where it doesn't work. I had a friend who, she had a Staphylococcus aureus infection on her face. So that's not fun, right? Having this big rash on your face. And I don't know how many different antibiotics she went through, but it took a couple of months to finally get rid of that thing. And of course, you know, no one wants anything like that on your face, right? So bacillus, aerobic, and spore formers. And the reason why I mentioned that one is because the other big group, and we're going to start talking about these ones in particular, are the Clostridiums. So that's another genre, and it includes at least 3 organisms we're going to talk about in this course. And these ones are anaerobic. And they're also spore formers. And then the last group, something we've talked about, is Listeria. And so Listeria, I'm going to underline that. It's a genre. So no spores for that one. So like I said, that's pretty easy to test for. And another thing about them is that they're motile. So there's different ways to test for motility. There's flagellar stains and things like that. And I'll show you some pictures of that in a moment. Okay, so I think we got all the gram positives. I was able to squish it on there. The gram negatives, I think I'm going to have to move over here. I don't know if I can pick that up. Somehow. There's got to be a way I can pick up the rest of that and make some space. Okay, so hopefully you got that for the gram positives. If you missed it, just let me know, or you can take a look at the video. I'm going to move the gram negatives way over here so I have lots of space for them. So here we go, gram negatives. Okay, so gram negatives. They stain pink and red, and they grow in the McConkey agar. So 3 common options now for the gram negatives. So first one are the diplococci. Okay, so we'll draw them, and they look like this. Sometimes the joke is we say they look like little bums. Or little snowmen, or whatever you want to think they look like. So there's the diplococci. Second, we can have bacillus-shaped cells. Okay, and so we've already drawn bacillus in purple. Now we're going to draw bacillus in red. Oh, there's a nicer shape. There we go. So sometimes they're kind of short and stubby rods like that. Sometimes they're a little bit longer. The same organism can grow long or skinny. Again, sometimes it just depends on the age of the culture or whether they're close to dividing and things like that. But, you know, E. coli can look like both of those things. And then the last one was Vibrio. So Vibrio, here we go. I'm going to draw some little bent rods. There we go. You wouldn't think this would be very hard to draw something like that. And they can look a little longer, a little shorter. That's kind of more typical what I drew, what you see. Okay, so what organisms are we talking about here? Okay, so let me see here if I can find a new color. I've just discovered very recently that there's other colors I can use on this. So it's kind of exciting. So diplococci, we've talked about both of these organisms already. We have, they're both Neisseria organisms. Neisseria, I'm just checking to make sure I spell that correctly. I tend to forget a letter or two once in a while. So we have Neisseria meningitidis, and the other Neisseria is gonorrhea. And I will never ask you to spell gonorrhea because it has so many weird vowels and whatnot in there that I never can get it right on first try. But if you can spell it, all the better for you. So there are tests to distinguish between the two of these. But usually it's where did you get it, right? Did you take this organism from a genital sample, so some pus found, from maybe on the tip of the person's penis or something like that, or did you get it from a spinal tap? And that usually tells you which one you have. Usually there's no other questions about that. Yep, we have a gram-negative Diplococcus from somebody's urine sample, they've got gonorrhea. You can do antibiotic tests. Neisseria is very good at getting resistance. It just likes to pick up DNA from the environment. So in the last maybe a decade or so, there's been a lot more resistance, and so sometimes it requires a second course and maybe some antibiotic testing. I don't know what the standard treatment is. It used to be just penicillin. I think now what they're doing is giving more broad spectrum antibiotics for that case. Okay, so bacillus. Let's see here. I'm just going to see if I can fit a little bit more on the screen. So unfortunately, this is the one that I'd have to say there are many. Way, way too many. And I'm going to list some of the ones that we've talked about. And give you a couple of things that might help you distinguish which ones we have. So we have E. coli, right? So Escherichia. We also talked about Yersinia pestis. That is the one that causes the plague. Hopefully you don't have that one. We also talked about Salmonella. And we've kind of mentioned Pseudomonas a few times, kind of in passing. And I kind of talk a lot more about Pseudomonas, Did I miss something? Yes, I did. I missed some letters. You know, it's a lot easier to spell when you're not talking and thinking all at the same time. Anyway, so many organisms fit into this category. If you have a gram-negative rod, right, you could have a whole bunch of different things. So there's all sorts of other tests you're going to look at. And those test kits I was showing with all those metabolic things that you're looking at are usually what are used at this stage, or sometimes DNA tests, right? Kind of depending on the context. Like I said, the clinical context can be huge in many cases. So E. coli, what can we say about E. coli? E. coli is motile. It has flagella. It's catalase negative. And I'm going to put one other test in here. Often people are looking at can this thing eat lactose or not? And that can be seen on different types of media. I also mentioned the EMB media where they have a really cool green color. So that's something that's looked at. There's many strains of E. coli, so there's quite a few different tests that can be done. But if you're trying to distinguish between E. coli and Salmonella and Pseudomonas, I'll show you what these tests look like. So all three of these, unfortunately, are motile. Yersinia, actually, I'm not 100% sure. I don't have a note for that, because usually we're not testing for plague. So these other two, though, are catalase positive. So you're going to see bubbles. And at this stage, I usually know whether it's Salmonella or Pseudomonas. Pseudomonas, the colonies are kind of a greenish color, and once in a while they're a purplish color, a very distinct, kind of interesting color. Usually you're not asking the question, do you have E. coli? You know, sometimes you might be looking between these three organisms, but that's an easy one to look at. Salmonella also usually forms a yellow color on the agar, the McConkey agar compared to E. coli. So another easy test to look for. The other thing, though, is that Pseudomonas is lactase negative, and there is a lactase test. It kind of looks like that urease test I was showing you, where you get a different color. You get I think it's a red-yellow test that you're looking at. Okay, so let's just zoom out here, see what we have for the gram negatives. Okay, so the last one there you can see is Vibrio, and one organism that we're just kind of mentioned a couple of times because it's a good example of a Vibrio that doesn't really come up too much in this class is the one that causes cholera. So this is called Vibrio cholerae. So like I said, I'm not really going to talk about cholera a lot. There has not been a cholera epidemic in Canada in, like, who knows, ever, maybe 150 years. But it is an important organism in many developing countries. Happens in India all the time. There was a big one that happened in Haiti after the earthquake a few years ago. So a significant organism, but in this case, I'm just saying, hey, it's a good example of a Vibrio. Somebody's asking about Yersinia. I don't know if it's motile or not. I'd have to look that one up. Usually you're not testing for it, thankfully. Okay, so zoom out. There's my massive flow chart. I guess I could extend this arrow all the way over here. It's a little easier to all fit on when you have a piece of paper, but drawing on the computer screen is not so easy to do. You can see all my different sizes in fonts on there. Okay. So I'm going to go back to the PowerPoint. Like I said, if you've missed that, you'll have to take a look at the video or let me know. I can probably export this and share it. And I can send it your way if you want to send me an email. So let's go back to our plates that we were doing some swabbing on. I didn't have a chance to do all the gram stains that I wanted to do. I think I was only able to do a couple. I just ran out of time. I had to do some gram stains for another class as well. But I'll show you, what I was seeing. So this plate here, if you look at this, I selected that colony there. I was hoping to do this one here as well. I can see it's a little bit of a different color, and this one here as well. So, maybe at some point I'll get a chance to do those, but not at the moment. But I'll show you that one there, what I got, the one circled in black. Gram-positive staphylococci. So probably no surprise there. I suspected that was Staphylococcus epidermidis. So these white colonies here, I was mentioning Staphylococcus epidermidis. It grows well in a petri dish. It's kind of not quite a white color, but close compared to the other colonies. It looks white anyway. So this is a gram-positive Staphylococcus. You can see they're purple. There is a bit of a red halo around them. I probably didn't wash it well enough or left the safranin on too long, but it's definitely gram-positive. It's dark. You don't get these kind of dark colors when you're dealing with a gram-negative. So my guess for this one is this is probably Staphylococcus epidermidis. Why am I excluding Staphylococcus aureus without doing a coagulase test? Because it's not yellow colonies. And like I said, that's a pretty easy one to tell right there. So what about the keyboard? I did circle this one. I was going for something large, and we have more Staphylococcus. Didn't turn out so well. My photo was a little blurry. I know, and it's the purple again. I'll tell you for this one, when it was in focus, it was more purple, and when it was blurry, it was looking more pink, which is just showing that safranin is all over the glass, and sometimes you see that kind of thing. But you'll see what the gram-negatives look like, and the pink is much, much more obvious. So that's probably more Staphylococcus epidermidis. So here's the toddler's hand. This was not actually this semester. I did this one last year, and it was a good one because I actually chose this kind of, that fuzzy colony there. And in fact, that was actually a gram-positive, so purple, and it's a bacillus. And if you look carefully at those cells, I'm kind of looking at this one here, for example. It looks like I probably have some endospores starting to form there. I'm not sure which bacillus this is, probably Bacillus subtilis. And why am I saying that? Because it's one of the most common ones that's found out there environmentally in the soil. And toddlers are all over the place, and try getting them to wash their hands. It's a big job. So what about the keychain here? This is not one that I did last year as well. And I chose this one because I was looking at this particular colony here, and I was thinking, hey, that looks a lot like E. coli. So I did the gram stain. Where's the gram stain? There we go. There's the gram stain. And I didn't do any further tests on this. Like I said, just a quick gram stain, but I know from looking at that colony, it looks a lot like E. coli. Maybe not as obvious from the picture. Usually it's kind of shinier. These pictures are hard to take. And I got these gram-negative rods. So probably E. coli there. Although, like I said, with the gram-negatives, there are many, many possibilities. It grew well in a petri dish. So, like I said, E. coli tends to be E. coli 9 times out of 10. Hopefully not salmonella on that keychain. So I wanted to show this plate with you here. I had mentioned I had these large petri dishes that I bought to put cell phones on. So this is an older picture. My colleague, he has a new phone now, but as of last year, he had an iPhone 4S. I know everyone's thinking, wow, that was a long time ago. Yeah, just last year he had an iPhone 4S, and he had this this case on it, and the case was kind of this textured leather case, and it was perfect. You can imagine just the texture in the leather capturing bacteria. And so I said, hey, can I borrow your phone? And he's like, sure. And so there's actually his phone right there, and you can see the outline and image very nicely. this fuzzy stuff here at the top, that was some sort of fungi that grew. And yeah, we got a nice picture out of this here. And so I tested that, and I got more Staphylococcus, Staphylococcus everywhere. And no surprise, every time I do this, I always find tons of Staphylococcus, because like I said, it's on our skin, it grows well in a Petri dish, and we're touching things all over the place. We're touching keyboards and mouse and doorknobs and all sorts of things. So pretty easy to do, like I said, a quick and preliminary, at least guess at the identification of these organisms just from a gram stain. So there's another one I wanted to share with you. I thought that was cool. It's not always easy to get both gram negatives and gram positives in focus on the same slide. So I was able to do it for this one. And you can see we have two different types of cells there. So that picture turned out really nice. And you can see in that case there, the gram positives, the contrast with the gram negatives, very different, the purple and the pink. Okay, so I kind of wanted to talk a little bit about some other microscopy stuff here. I'm just looking at the time here. We got about 10 minutes, so we'll get through some of it. Maybe we'll have to save the electron stuff for next day, but we'll see. So we're talking about cell walls a while ago when we're talking about bacteria, and I mentioned that not all of them are gram negatives and gram positives, just kind of most of them. The exception to the group and the medically important one for this class is tuberculosis. So mycobacteria, if you remember, I showed you this picture before. They have these mycolic acids, and these mycolic acids are very waxy, and the gram stain doesn't stain them. So the crystal violet and the saffron, it just doesn't get inside. It's waxy, so it washes off way too easily. So sometimes people call these acid-fast cell walls. And so you can see that name there, acid-fast. That has to do with the staining process. So I want to talk about this staining process as well for acid-fast organisms. So what does acid-fast mean, right? So there's an acid in there somewhere. You probably realize that. The acid is that decolorization of the washing step. And fast doesn't necessarily, in this case, mean speedy. Fast means, like if you ever heard somebody say the expression, hold fast, right? It means hold tight, right? And so fast means resistant or something like that in this particular case. You can see this stain has another name. It's called the Zeil-Nielsen stain. Most people say acid fast, but once in a while you'll see either term. So let's take a look at this, and I want to kind of, oh, there's my explanation. Acid means acid, fastness means secure or resistant. So I'll show you how this one works, right? So the first stain that you throw in is this carbol fuchsia. It's also actually called fuchsin, but I call it fuchsia because that reminds me of what the color is. So you probably know what fuchsia is. I know lots of people with all sorts of fuchsia garments, phone cases, and purses, and once in a while you see someone with fuchsia shoes or pants. It's that bright, strong pink color, and we require lots of heat here. So we need the heat to melt that waxy mycolic acid stuff. So we're talking about concentrated chemicals, lots of heat, and we're trying to drive the stain in. So this will get everything. Everything gets stained at this point. The next step is you're using an ethanol of some sort with acid in it, and again something relatively concentrated because you're trying to decolorize things. Now the acid-fast cells, like the tuberculosis, that mycolic acid has solidified and the acid just doesn't get in. Other cells, the acid gets in and it wipes away the colored cells. So now you have kind of a similar situation what we saw before. You have you know, something that's colored and something that's not colored. So you throw in a counterstain, and this counterstain is methylene blue. Methylene blue is used for all sorts of things. It's just kind of a general stain used for yeast and other things, and it's a light color. And so the fuchsia is such a strong dark color that you don't see it, the methylene blue. So I'll show you some pictures. So there's a picture there. This one I found off the internet, but you can see in this case here the methylene blue is darker, so I kind of wanted to show you that. And you can see there's a contrast, but usually what they look like is something like this. And so what you're seeing in the background, this is probably from a sputum sample. And so these things in the background, these big blue things, those could be fragments of human cells. Human cells are a little bit bigger than that. So that's why I'm saying fragments of some sort. And then you've got these little rods here. So they're little bacilli, and they're staining that fuchsia color. And so that's a pretty classical kind of diagnostic test for tuberculosis, is staining a sputum sample and looking for those rods. I think I have another picture. There's another one, and you can see those rods are all over the place. So there are a few other stains out there. And I mentioned, for example, that Streptococcus pneumonia has capsules. So there's a capsular stain, and you can see in the capsule stain, what you're doing is the stain, it doesn't permeate the capsule very well, or the slime layer. So you've got, you know, that goopy layer around the cells, and it kind of looks like these little halos. So the stain will get on the slide, it gets on the cells, and then you have these halos around it. I've never actually done a capsule stain. Apparently they're pretty easy to do if you're looking for that kind of thing, but they're not necessarily done routinely. There are also flagellar stains. So flagella are very skinny. So you need special stains for that. You don't see them under a gram stain. You need actually a kind of stain that makes them thicker. And so it binds to the organism and makes the organism thicker, but as a consequence, it also makes the flagella thicker, and then you can actually see it. Otherwise, you'd never see it under, like using a normal stain. There are special endospore stains. So I showed you my bacillus there, and I saw evidence of endospores. There are some stains that I'm not sure what they're binding to, but some molecule that's in the endospore and show up a different color. So there's an image there for you from the textbook. Okay, so I have a couple minutes left, and that's probably just enough time to talk about fluorescence microscopy, and we'll leave the electron microscopy for next day. So what is fluorescence microscopy? Well, you're using, it's basically a special kind of stain, and this stain will glow. So that's what fluorescence is. I can talk to you about the physics if you want, but I'm not going to get into it. But basically, you shine a light on it, and it glows a nice color. So now you can get your specimen glowing, and it's going to actually, you're improving resolution a little bit, and you are, you know, it's very, very visible. You can see I have this other type listed here, confocal microscopy. So confocal microscopy is basically a more expensive type of fluorescence. So a fluorescence microscope for a basic one is $20,000, right? So, you know, that's expensive. I'm not going to buy one for my house. But, you know, a hospital might have one or two, right? Because they find them useful. Confocal microscopes, I think a cheap one is $50,000. I think most people are spending $100,000 or $200,000 on them. And so what's different about them? They have lasers, and these lasers, you can focus things very precisely, and usually they come with really expensive software as well, and you can do some fancy things with them. But it's basically a type of fluorescence microscopy. So I'll show you some pictures. Your samples are glowing. You got really wonderful contrast between the background and the actual cells. So there's some Staphylococcus aureus. It's not purple because we're not staining it with a crystal violet. We're staining it with some sort of green thing. What else do we have here? There's lots of beautiful pictures of fluorescence microscopy that you can find if you just Google on the internet. You can see some cool things. In this case, you can see they've used two different stains. One stain is staining something in the nucleus, and the other stain is staining something in the cytoplasm. So you're getting some really cool images from this. There's Giardia, and I'm not sure what the red things are. They're probably ribosomes, and you can see the two nuclei. So very cool. Like I said, somebody's got to make a science fiction movie about a giant Giardia. It'll definitely be scary. There's another one there. You can see somebody's looking at spindle fibers and chromosomes. So very, very cool stuff. Lots of pictures to show you. This one here, I don't know what they're doing, but you can see there's a worm there. Again, I can't remember what they're doing on this one, but somebody made a little rainbow on this cell, and I thought it was just kind of interesting to look at. So fluorescence and confocal microscopy. So remember, like fluorescence, like I said, you know, it's not quite as good as confocal. And if you have thick samples, it's kind of like think about your eyeballs focusing. If you're focusing at a piece of paper in front of you, your eye is also not focusing at the car across the street. If you're looking at the car, you're not focused here. It's the same with microscopy. If you have big things like eukaryotic cells, they're thick, and you can't focus the whole sample at once. You can just focus in one part. So that fluorescence on the left, that's what's happening. They're trying to focus, and some things are going blurry, and other parts are going focused. Confocal can shine that laser in at precise depths, and you can focus just part at a time and kind of reconstruct the whole thing in the computer afterwards. So that's why it's a lot more expensive. I'll show you a few more pictures here, and then we'll wrap up for the day. Some other cool things here. I don't know what they're doing here. This is from, I can't remember, one of these photography contests, Nikon or National Geographic or something. And people submit these images, and it's just like, well, that's just incredible. Here's another one you can see called Up Close and Personal. This is same contest. You can see the nucleus is just popping out at you, and all those skinny little yellow things are mitochondria. Very, very beautiful images. Here's another one. This is this one, Nikon. This is the first place in 2017, and that's a skin cell. So very cool pictures. I'm sure they have very expensive microscopes. So keep in mind, right, you know, we're thinking about diagnostic techniques, right? So here's somebody looking for Plasmodium. So if you are a technician and you're looking for, you know, somebody, do they have malaria? Yes or no? What they do is you take a blood sample, gets smeared across a microscope slide. And traditionally what they would do is there's a stain, you put it on there, and the technician is trained to know what to look for, which is great. But sometimes you get false positive, false negatives. With fluorescence, we can eliminate a lot of that. It's very, very obvious that you have these yellow things in this image. And those yellow things are the Plasmodium parasite. So it's a lot more expensive, but you're getting much better results. So, you know, there's that trade-off with a lot of things. So in some cases, we're starting to shift to more fluorescence diagnostics nowadays because of the ease of use and the speed can save you money as well. Just wanted to show you a couple more pictures before wrapping up. I mentioned with the confocal microscopy, you can focus in at different depths. So kind of a cool thing that you can do is actually build a three-dimensional image. So that's what they're doing here, is they're not showing you the colors, but they're taking the information from the image and actually building a three-dimensional image there. So here's another picture. This is Mycobacterium tuberculosis. You can do the acid-fast stain. It takes takes a few minutes, or you can do the fluorescent stain. And the problem with the acid-fast stain is, you know, sometimes you get all this mucus and debris, and you can't see the cells. Under fluorescence, much, much easier. So like I said, we're kind of moving towards fluorescence in a lot of cases as the prices are coming down, and you know, if it saves you a lot of time, even better. Okay, so I think that is where I will finish for today. So I do have a couple other things to say about fluorescence, but we're kind of running out of time, and we are out of time. And we'll talk a little bit of electron microscopy, immunological things, tests, and DNA tests next day, and we might start into the following topic, depending on how much I actually have left. Okay, so that is all for today. Hopefully everybody has a great long weekend. I know we could use a long weekend every weekend, but so savor them when you get the chance. So take care, and we will see you next week.